significant correlation in adult rain between the concentration and the number of straws, as well as for the volume of ejaculated with the values of the number of straws. Statistical analysis shows to existence in young rains of a significant correlation between the concentration and the number of straws. Principal component analysis. According to the principal component analysis, the ascending hierarchical classification is represented by first group, represented mainly by the winter and summer season. This group is negatively correlated with the factor studied. It is characterized by low season activity. Second group, represented by autumn and spring, during which we are recording an effect of the season which is quite strong and which correlates positively with the factors represented by strong season activities. The age category effect of rains has been shown to be a factor of variation as quantitative semen production is greater in adult than in younger. This difference is very important for the mass motility and significant for the concentration, but not significant for the volume and the number of straws. The quantitative parameters of regular rain semen are subject to seasonal variation. Unlike winter and summer, the best profitability of rains is the autumn and spring period, when the ejaculate volume, concentration, and number of straws of rains are greater. The effect of season has a highly significant influence on the volume compared to the other three parameters: concentration, number of straws, and mass motility.
Thank you very much, Mr. Dr. Ahmed Benya, for your presentation. And about the present here, if they have any questions for Ahmed Benya about this presentation, please. If there is no question from the presentation here, we have to send the word to Madam Dr. Baha, Munya, or Ben Aziza to pose some questions about this.
Merci beaucoup, Monsieur Kharoubi. Ce qui concerne l'intervenant, j'aimerais bien savoir, c'est-à-dire pourquoi comment vous expliquez les variations saisonnières. Merci pour votre réponse.
Bon, bonjour, salam alaikum. Donc, en ce qui concerne les variations saisonnières, elles dépendent beaucoup plus de l'effet de la photopériode. Ça veut dire de l'effet de la longueur du jour. Donc, l'activité sexuelle chez les ovins et les petits ruminants d'une façon générale est plus élevée là où les jours sont courts. Par contre, dans notre étude, nous avons constaté que cette activité chez les béliers de la race ou les tchellèles, elle dépend beaucoup plus de l'alimentation que de l'effet de la photopériode. Ça veut dire que les saisons où les disponibilités alimentaires et fourragères sont bien présentes, l'effet de la photopériode est masqué tout simplement.
Merci. Et je tiens vraiment à savoir, enfin c'est un très bon travail, et puis je tiens à savoir, est-ce que vous avez pratiqué ce même type d'expérimentation à d'autres animaux? Et puis je donnerai directement la parole à ma chère collègue, Madame Benaziza.
Donc, on est en train de travailler actuellement pour d'autres races, toujours pour les petits ruminants, à savoir la race Rambi dans la région de l'ouest d'Algérie.
D'accord, merci. Donc, je donne la parole à professeur Benaziza.
Merci beaucoup. Merci à M. Ahmed
Merci.
Ce que je voudrais savoir, donc vous avez essayé de... Est-ce que vous avez essayé donc de montrer, vous venez de dire que c'était beaucoup plus l'alimentation. Donc, vous avez soumis vos animaux à un flushing, je pense.
Bien sûr, bien sûr.
Voilà, un flushing. Et est-ce que ce flushing-là, il consistait en quoi?
Vous pouvez répéter l'action?
Pardon, je disais que votre flushing, il consistait à donner quoi?
Une ration, un concentré commercial à base de beaucoup plus 40% de d'orge, 20% de maïs, 20% de soja, ainsi que de CMV.
C'est un flushing qui est normalement constitué à plus de 50% de produits, c'est-à-dire de matières premières qui sont importées. Hein.
Importé beaucoup plus, oui.
Le soja aussi. Est-ce que vous n'avez pas essayé?
Le maïs, soja. L'orge, c'est l'orge de notre pays. Par contre, le maïs soja, c'est des produits importés.
Le maïs et le soja sont importés. Très bien. Est-ce que vous n'avez pas essayé de remplacer le soja, par exemple, ou bien le maïs par d'autres matières premières?
Non, on n'a pas essayé ça.
D'accord, d'accord. Parce que si on parle toujours d'importation, vous savez très bien que maintenant, pour l'importation, donc si le flushing est soumis donc à des matières premières d'importation, ça va nous revenir un peu plus cher. Donc, moi, ce que je vous conseillerais, c'est d'essayer d'utiliser le flushing en utilisant d'autres matières premières, des matières premières locales, quoi, c'est-à-dire pour une expérimentation. Voilà. Je vous remercie.
Merci bien, merci madame. Merci.
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 Donc, Monsieur Kharoubi et Monsieur Benzahra, on vous redonne la parole pour le prochain communicant. Merci.
Yes, thank you very much for your notes, your remarks, and intervention. So we have quickly to the second presentation about the ID number 3. I don't know if he's here present or no. ID number 3. Okay, we can start with the number. ID. Yeah, okay.
Yes, we are here.
Okay, thank you. We can start.
Okay.
Assalamu alaikum. Hi everyone and welcome to my presentation. First of all, let me thank you all for attending this very important event. Atlas pistachio is a rustic species with a great plasticity in different ecological areas. It has an agronomic interest as a graft holder for Pistacia vera, industrial for its oils, and ecological thanks to its pivotal and strongly branched root system. The Algerian steppe are subject to desertification and overgrazing, which prevent the reconstitution of steppe rangelands. In this context, a new approach is proposed for the rehabilitation and repopulation of these degraded rangelands by using this endangered species, which constitutes, according to reforestation program, an adequate solution. In nature, the regeneration of Atlas pistachio seeds, the Atlas pistachio seeds, is very weak, for they are characterized by mechanical dormancy with their teguments and biological with their content of polyphenol and fatty acids. Our study is focused in this context in order to improve the germination rate of the Atlas pistachio seeds by proposing a pre-treatment. In addition, a study was carried out on the genetic variability of 4 populations by the ESSR molecular marker. For the sampling, seeds of Pistachia atlantica were collected from 10 trees in each of the 3 locations in Algeria, Batna, Bishar, and Jilfa, from the beginning of October to November 2015. To study the seeds germination, the seeds of Pistachia atlantica were divided into two groups, with epicarp and without epicarp, which was achieved by mechanical scarification. The seeds were imbibed for 48 hours in distilled water for a control, or 4% H2O2 as a pretreatment. For the second part of this study, genomic DNA was extracted from leaf tissue by setup method with the modification adopted with KIFCAS of 2006. For the ESSR PCR amplification, the samples were genotyped for ESSR markers using GC and CA-rich primers. Our results showed that the germination rate of manually scarified seeds was 0. On the other hand, the application of H2O2 induced a significant increase in the germination rate of seeds. Biochemical analysis of the different parts of the seeds, epicarp and kernel, showed their richness in total phenolic compounds, which induces the biochemical dormancy as well as tegumentary dormancy in these seeds, and consequently an inhibition of germination. H2O2 improves the germination rates of seeds by oxidizing the inhibitors in the epicarp and kernel and releasing oxygen, sterilization by elimination of pathogen, maceration by increasing permeability of tegument to oxygen and water, nutritional by giving a potential substrate to several enzymes. The study of the genetic variability of the 4 populations by ESSR molecular marker showed that their clustering was related to the geographical locations and the climatic conditions of these regions. The assessment of genetic variability by ESSR confirms the genetic richness and distribution of Atlas pistachio across Algeria. To conclude our presentation, we can say that our results show that the protection of this species requires the mastery of the reproduction methods, particularly sexual. Based on this study, we were able to prove that the tegumentary and embryonic dormancy of the seeds greatly limits the sexual reproduction by seed sowing. Based on the phylogenetic study, we were able to consider that for the reproduction of this species, it is important to proceed in the future by the valorization of the genetic variability. Finally, it is crucial for the repopulation operations in different regions to be carried out using seeds collected from the local ecotypes. And thank you.
Thank you very much, Michel. So quickly we have to pass the word to our professor, to Madame Daha or Madame Ben Aziza, if they are here. Madame Ben Aziza. So, she's here.
Salam alaikum. J'ai perdu ma connexion sur mon portable. Voilà, donc, je vous en prie. Voilà, donc j'aimerais bien savoir en ce qui concerne la méthodologie expérimentale. Est-ce que c'est une méthodologie qui est récente parce que là, il y a d'autres méthodologies qui ont été appliquées pour ce genre de travaux? Et puis, tout simplement aussi, je tiens à vous remercier énormément du travail que vous avez fait en choisissant ces 3 régions, en sachant que le travail sur terrain est très difficile. Merci pour votre réponse.
ID number 3, if you are hear me. Il Abdelli Amina ou bien Tahirin Mohamed. if you are here, we have some questions about your work.
Bon,
No.
je pense pour ne pas perdre du temps, on va passer à la prochaine communication.
Okay,
allez, on
we have to start with the next presentation.
Yes.
About the ID number 36. 36 is Naima Bilgage and Mohammed Masnoua. If you are here, ID number.
Yes, I'm here.
Yeah, okay.
Yes, I'm here.
Yeah, so hold on with us. You can stay with us because we have some question about your work after your presentation, please. Okay? So we can just start now.
From scientific and morphological characteristics of some Algerian date palm fruit varieties from the Sahara oasis. The Algerian date production of the 2008 campaign was 5 meters of all varieties, 8 million in 2012 and 9 million meters in 2013. The potential for date palm increased from 11 million in 2000 to 80 million palms in 2030. The very important genetic diversity of the date palm in Algeria, all about 1,000 varieties. With the economic and social evolution of Algeria, the palm groves of different oases are being reorganized to meet an ever-increasing demand for superior quality dates like the elite cultivar de Glenmore. As a result of this reorganization, vinicultors have moved from a rich and diverse traditional cultivation system to an industry system focused on monovarietal cultivation. It's therefore imperative to develop various actions aimed to safeguard its plant genetic resource. From this perspective, varietal identification by exploring the physico-chemical potential of the fruit of each cultivar constitutes one of the essential compounds for rational management of each viticulture heritage. Material and methodology. First of all, for the plant material, we have studied 7 different date palm varieties, which is Adam Bulla, Hmira, Kahlaya, Fagus, Sherka, and Tiglit. They were harvested from Sahara oases located at Adrar, Algeria, and stored at 4 degrees for further analysis. For the characterization analysis, we have 1. The morphometric analysis like length and width of the date, length and width of the peat, total weight of the date, pulp and peat, and pulp and date ratio, and peat for date ratio. For the physico-chemical analysis, we have the moisture, pH, travel acidity, total sugar, reduced sugar, total H's, minerals, total phenolic compounds, and total flavonoid contents. For the statistical analysis, we have the analysis of variance ANOVA and to the correlation versus coefficients. All parameters were determined in triplicate for each sample. Statistical analysis were performed with Excel Stat version 2009. Analysis of variance ANOVA and two-case test at the threshold of 0.05 was used to determine significant differences between means. Correlation analysis was performed employing Spearman's test. Results and discussion. First of all, richness in pulp. The date varieties studied are generally rich in pulp. with an average per 4 days ratio equal to 0.88 with a standard deviation of 0.08. These results are close to that of Huda 2014, where the 30 varieties of Moroccan dates studied have a per 4 days ratio of around 0.8 to 0.9, while the 7 Algerian varieties studied have a ratio varied between 0.5 and 0.9. In addition, there is no significant differences between them at the threshold of 0.05. For the master, according to the classification of the varieties of dates studied, they are generally soft in texture, with an average of water rate of 26% with a standard deviation of 5. It varies between 11 and 33%. Hence, there is a clear difference between the 7 varieties studied at the threshold of 0.05 with M squared equal to 0.96. These results are similar to those of Jordan, who worked on 8 Algerian varieties from the region of Garbin, where the humidity rate varies from 11 to 39%. For the total phenolic compounds, the dates varieties studied are generally rich, with an average of 57 mg of gallic acid equivalent for 100 g of fresh fruit and a standard deviation of 26. The polyphenol total compounds vary between 12 and 109 mg of gallic acid equivalent for 1 g fresh fruit. And there is clear difference between 7 varieties studied at the threshold of 0.05. These results are clearly much better than those of Mansouri and Alimi 2005, where the total phenolic compound of 7 other Algerian from the Gardayam Zab vary from 2 to 8 mg gallic acid equivalent for 100 g of fresh fruit. In comparison with other studies, it's found that the level of total phenolic compounds in dates differ from variety to variety and from region to region. The quantity and composition of the physical chemical present in dates vary widely depending on the dates variety, storage, post-harvest processing, experimental condition used for the analysis, and the geographic origin of the dates. For the total sugar content, the date varieties studied are generally rich in sugar, with an average of 14 mg of glucose equivalent for 1 ml of extract, and standard deviation of 7.1. The sugar level varies between 6 and 26 mg of glucose equivalent for 1 ml of extract, from where there is a clear significant difference between the 7 varieties studied at the threshold of 0.05 with an R square equal to 0.98. Many authors, including Mini, P, and Alimi, who have worked on several varieties of dates, claim that the sugars of dates vary according to the variety. Regarding the correlation, we have found three types of correlation. First of all, correlation between the morphometric parameters in themselves, 2, between morphometric and physicochemical characteristics, and 3, between physicochemical characteristics in themselves. First of all, the correlation between morphometric and physicochemical, we will see the main correlation. For example, we have a negative correlation between the date length and the total flavonoid with R square equal to 0.92, and a positive correlation between the bit white and the total flavonoid with the R square equal to 0.62, and a positive correlation between the pulp weight and hydration sugar with a 0.5 and a positive correlation between pulp weight with the total flavonoid with R squared equal to 0.9. For the correlation between the physicochemical characteristics in themselves, for example, we have correlation, negative correlation between moisture and reducing sugar, and positive correlation between tetrahydral acidity and reducing sugar. We did not find any previous studies on the correlation between the physicochemical parameters of the date, but according to Lane 2012, who investigated the correlation between quality attributes of Elchi genotypes, it was found that tetrahydral acidity was significantly and negatively correlated with reduced sugar with R squared equal to 0.5. And we have found that humidity is inversely correlated with the rate of reduced sugar with R squared equal to 0.9 at the threshold of 0.5. For the morphometric and morphometric correlation, we have the date length is positively correlated with the seed length and the date length with R squared equal to 0.8 and 0.5 respectively. The correlation between the date length and seed length is positive, but the correlation between the fruit length and seed weight is very weak, although significations, partly due to the large range of the variation in fruit weight. According to Ghanimi, the irregularity of the date fruit shape explains the weak relation between fruit weight and the fruit and seed dimension. Generally, the shape of ripe date fruit varies from spherical to ovoid or cylindrical with 3 or 7 centimeters length and 2 to 3 centimeters of diameters. Conclusion. This study provides an overview of the characteristics of date palm varieties. The samples of these date varieties showed significant differences between the morphometric parameters in themselves, the physicochemical parameters in themselves, and between the morphometric and physicochemical parameters. We can conclude that there is a potential diversity in quality attributes of date palm fruits obtained from the Sahara oasis. The data obtained from analysis in this study may help in expanding the utilization of this date palm fruit in agro-industry. And thank you for your attention.
Thank you. Thank you very much for your presentation. So, about the audience, if you have any question about this work or presentation.
Oui, Monsieur Khalobi, je crois qu'il y a Madame Zouari qui est aussi avec nous ici présente, qui veut poser quelques questions. Donc, je lui donne directement la parole.
Ok, Nafila.
Bonsoir tout le monde. J'aimerais bien poser une petite question. Justement, sur les résultats que vous avez donnés, j'aimerais bien savoir, surtout pour les caractérisations physico-chimiques des dates, quelles sont les dates qui ont un meilleur paramètre pour la santé, qui présentent une meilleure nutrition.
Bonsoir.
Bonsoir.
Madame, vous m'avez demandé quelles sont les variétés de dattes qui présentent le meilleur amendement élément nutritif. Bon, pour pouvoir répondre à cette question, il faut faire beaucoup d'analyses, premièrement des vitamines, deuxièmement des oligo-éléments, les micro-éléments, ainsi les les acides aminés et beaucoup d'éléments nutritifs. Nous, on s'est intéressé plutôt, selon les moyens, aux sucres totaux, aux sucres réducteurs, glucose et fructose, et aux éléments minéraux, magnésium et sodium et potassium. Donc, ça ne se suffit pas. Deuxièmement, en Algérie, il y a à peu près 1100 variétés. Donc, moi, j'ai juste travaillé sur cette variété pour pouvoir, cette variété de la région de Saoura, Adrar. Donc, je peux pas dire que... Donc, pour les variétés, il y a une multitude de variétés, et chaque variété a des caractéristiques physico-chimiques et morphologiques distinctes. Il y a ceux qui sont riches en polyphénols, ils sont ceux qui sont riches en sucre, sont ceux riches en protéines. Et ces variétés, à part la variété d'aigle noir qui est la plus appréciée et la plus consommée, mais il faut aussi faire une exploitation physico-chimique des autres variétés puisqu'elles sont très intéressantes pour l'utilisation en agro-industrie. C'est-à-dire, on peut exploiter ces caractéristiques physico-chimiques et morphologiques et par exemple la couleur et la texture pour l'utilisation en industrie alimentaire, pour le conditionnement et la transformation, par exemple pour le jus. Pour la fabrication du sirop ou bien le jus de date, on peut choisir des variétés riches en sucre, par exemple. Mais pour savoir quel est l'élément ou bien quel est le principe actif le plus comment dirais-je, pour savoir qui est la variété la plus nutritive, il faut un travail plus large. Voilà.
Je dois vous remercier, madame. Je donne la parole directement à madame professeur Benaziza et puis à madame Bouhelel Sarah, vous êtes là? Donc, je donne directement à madame Benaziza.
Oui, bonjour, merci. Salam alaikum. Dites-moi, pour les sucres, vous avez dosé les sucres totaux, vous n'avez pas eu l'idée de doser quels sont les sucres que composent ces dates, c'est-à-dire voir du point de vue diététique, c'est ce que a posé ma collègue maintenant la question. J'aurais aimé connaître au moins quelle est la part, par exemple, du fructose ou d'un autre sucre dans ces dates-là.
Les dates contiennent à peu près de 40 à 98% de sucre. Les trois éléments majeurs sont le glucose, le fructose et le saccharose. Il y a trois principaux groupes, on peut dire trois principaux groupes de classes de dates selon la composition en sucre. Pour les dates riches en sucre réducteur, sont les dates molles. Pour les dates riches en saccharose, sont les dates sèches.
Je suis d'accord avec vous, madame. Vous savez que maintenant, on fabrique de plus en plus de robes et c'est pour ça que je pose la question. Les gens ont tendance parfois à utiliser n'importe quelle date et ce que j'aurais trouvé la part de chaque sucre dans ces dates. C'est pour ça que le collègue avait posé la question de nutrition, c'est-à-dire du point de vue nutritionnel, quelles sont les dates qu'on peut utiliser le plus, c'est ça. Voilà. Je vous remercie, madame. Et je repasse la parole à madame Sarah. Voilà, madame Sarah.
Oui,
je suis là. Je vous remercie.
Merci beaucoup.
La parole est à madame Sarah Bourrel.
Oui, je suis là. Merci beaucoup. Donc, en fait, je pense que la discussion va se poursuivre par rapport au sucre. Donc, vous avez parlé
Est-ce que vous m'écoutez, s'il vous plaît? Très bien. Donc, merci. Alors, la discussion va se poursuivre par rapport au sucre. Donc, vous avez parlé de la valeur nutritive, etc. Est-ce que vous avez fait le dosage d'un point de vue quantitatif, bien évidemment, ou bien aussi d'un point de vue qualitatif? Parce que lorsqu'on parle de sucre, faut savoir qu'on n'a pas toujours une pureté satisfaisante. Donc, est-ce que vous avez pensé à analyser qualitativement vos molécules? Et merci.
Merci pour cette question intéressante. Donc, pour ce travail, c'est juste une analyse quantitative des sucres totaux et des sucres réducteurs. Pour les sucres réducteurs, les dates, ce sont glucose et le fructose sont les deux éléments majeurs pour les sucres réducteurs. Il y a aussi le maltose, l'inuline, comme présentent des quantités infimes. Donc, pour la caractérisation qualitative
Très
des
bien,
sucres,
mais
on doit
s'il vous plaît,
procéder.
pensez à vérifier la qualité. Parce que s'il y a, par exemple, si vous avez, je ne sais pas moi, 1% ou bien 2% de protéines, croyez-moi, ça change tout.
3% de protéines et 1% de matière grasse, voilà. Et 2%
Et voilà,
de
3% de protéines, c'est énorme, hein. Mais bon, ce n'est pas un souci. Merci pour votre
Oui,
réponse.
pour la caractérisation qualitative, on doit
Voilà,
procéder à
exactement.
l'analyse par HPLC et ou bien
Très bien.
CPG.
Merci de votre réponse.
De rien. Merci pour votre question.
Thank you. If there is any questions from the audience?
Oui, j'ai une petite suggestion.
Could you discuss?
Oui, c'est à propos du mot variété pour les.
You can start, yeah.
Oui, oui, c'est pour le mot variété pour le palmidatier. Je pense que le mot variété, le palmidatier ne respecte pas la définition botanique et agronomique du palmidatier. Parce que il doit être DHS, distinct, homogène et stable. Mais pour le palmidatier, on utilise le mot cultivar que le
Cultivar, oui.
variété. Voilà, exactement.
Oui,
C'est pourquoi.
agronomiquement parlant, on dit cultivar, pas variété.
Exact, voilà, exactement.
Pour le domaine de l'agroalimentaire, on dit les variétés de fruits, c'est tout.
D'accord, ok.
Oui.
Okay, thank you, Mr. Saad Wa'azza, for your attention. So, there is no question about this topic. We can start with the new presentation. Okay. About the ID number 48. Untitled investigation of in vitro antioxidant activity for shares of oregano, Hamala, plants growing in the semi-arid climate, presented by Amanate Marzouk, if she is here.
Yes.
Okay, so we can start.
Good morning, ladies and gentlemen. I would thank the organizers for giving me this opportunity to present our work in this conference on environmental biotechnology and biodiversity. My presentation will be on investigation of in vitro antioxidant activity for seeds of Peganum harmala plant growing in semi-arid climate. So we begin with an introduction and goals. Since ancient times, many plants have been used for the treatment of many diseases related to the toxic effects of oxidants. Oxidative stress is responsible for several diseases such as cardiovascular disorders, Alzheimer's disease, and cancer. The synthetic antioxidants can induce many side effects. For this reason, it is necessary to replace them with natural antioxidants. As one of the most important secondary metabolites, phenolic compounds are widely investigated in many medicinal plants. Among of them, Peganum harmala, which belonging to the family of Zygophyllaceae, traditionally used as an immunogogue, abortifacient, analgesic, and anti-inflammatory agent. For the objective of this work is to evaluate the antioxidant activity and the quantification of its secondary metabolites. So, our research is based on 3 steps. Firstly, extraction of secondary metabolites, then determination of phenolics and flavonoids compounds contents, and finally, the evaluation of antioxidant activity of chloroform extract with different methods. We pass now to the part material and methods. Total polyphenols and flavonoids were determined by Follen CiO culture and aluminium chloride methods. Regarding the antioxidant activity by DPPH, ABTS, reducing power, and ferrous gelatin, these tests were determined using the methods of Blois in 1958, Rui in 1999, Oizo in 1986, and Diniz in 1994. Firstly, the results showed that the chloroform fraction recorded a high amount of total phenolic and flavonoids compounds with values of 103.41 and 44.95 respectively. For the antioxidant activity of chloroform extract, the fraction showed a powerful antioxidant activity in DPPH, ABTS, and reducing power, but lower compared with antioxidant standards. However, the ferrous iron gelatin assay, the chloroform extract showed being no active. For conclusion, in the light of the results obtained concerning the contents of phenolic compounds and the antioxidant powers, we can conclude that this plant could be considered as natural sources for the therapeutic purposes. And thank you for your attention.
Thank you.
And I'm here for any questions.
For your presentation. So, about your work, we have some questions and interventions from the audience. Firstly, we have a question from Nafila. If you have any question, you can start.
Yes, thank you. J'aimerais bien, merci pour votre exposé. J'aimerais bien savoir, est-ce que vous avez comparé l'effet antioxydant de votre plante par rapport les antioxydants de référence qui est le BHA et le BHT. Et quels sont
Oui,
les résultats que vous avez obtenus?
Oui, madame, j'ai travaillé au niveau de centre de recherche de la biotechnologie.
Hello? We can't hear you. If you can repeat, please. We can't hear you clearly.
Do you Hello?
We are hearing you. You can start and talk, please.
Oui, j'ai comparé les résultats.
Amal, Amal, we can't hear you.
Oui, madame, est-ce que vous m'entendez? Madame,
Yes.
est-ce que vous m'entendez?
Oui, yes, oui.
Donc, j'ai comparé mes résultats avec des standards de référence comme le BHA, le BHT et l'acide ascorbique. Je l'ai mentionné au niveau de tableau numéro 2.
You have a bad connection, I think.
Monsieur?
Oui, I am hearing you. You can start, yeah.
Donc, j'ai comparé mes résultats avec des standards de référence. Donc, je l'ai mentionné dans le diapo, le BHA, le BHT, l'acide ascorbique et le EDTA.
Oui, justement.
Oui, justement, vous l'avez mentionné sur le tableau, mais par rapport à, je veux dire, par rapport à ces standards de référence, quel a été vraiment l'effet antioxydant de votre plante par rapport à ces plantes-là? C'est vrai que vous avez mentionné sur le tableau.
Oui, j'ai trouvé...
De point de vue comparatif.
Oui, j'ai trouvé une bonne activité antioxydante par rapport au standard. On ne peut pas dire que c'est une forte activité, mais une importante activité par rapport au standard.
Je vous remercie. Merci beaucoup pour votre réponse.
Merci, merci. We have another intervention of Mr. Saad Bouza. Mr. Saad. Mr. Saad, are you here? No? So, I have to thank Nate for your presentation and your answer.
Merci.
Okay, thank you very much. And we can pass to another presentation. And the ID number 61, yeah. Entitled Annual Land Cover falls under ICEBB classification using land time series. Cause the transition zone of Tierp region, Algeria, presented by Yasmina Amroun. If you are here,
Mrs. Yasmina Amroun. Hello? Yes, it's
yeah, okay. So we can start with it. Yeah.
Ladies and gentlemen, good morning. I'm Amroun Yasmina, currently PhD student at the University of Ibn Khaldun, Tieret. I'm here to present part of my research, annual land cover classification using Landsat time series. in the transition zone of Tielt region. I would like to thank the organizer to provide me with an opportunity to present this research. First, we have to talk about some background information. And if we can see here in this title, we have three terms, land cover, classification, and the transition zone. First, we have to ask what does mean land cover. Land cover usually is described as a set of hierarchical classes which describe the surface cover on the ground. And the data of the land cover can be presented spatially and statistics forms. So this is very important for many applications as crop yield estimation or forest management. Also, it's so easy to generate land cover maps using remote sensing data, but ensuring the accuracy is so difficult. The first step to get this land cover maps, we have the classification. As we know, it's a challenging task for many reasons. First, we have the complexity of the landscape, I mean the study area, also the type of remote sensing data, image processing, and the classification process. So, in general, the classification is the extraction of the information classes from multi-band raster images. Also, it's used as a discriminatory quality measure of the spectral and temporal bands of the images. For the method for the approach, we have classical and advanced methods. We choose classical, we have supervised and supervised, parametric, non-parametric, hard and soft, contextual based, and knowledge based. For data, remote sensing data, we have to know that each remote sensing data has some characteristics as a spectral characteristic, radiometric, spatial, and temporal resolution. Also, the selection of the suitable sensor data depends on many.
Please, Amel Naid, please close your sound or your micro, please. Thank you. Thank you. You can start again.
Factors as availability of virus images data, the characteristic of study area, the characteristic of the data like cost and time, and we have two important factors as scale and the resolution. So we have three types of scale: thin scale, used at a local level with the high spatial resolution, also regional scale with the medium used with medium spatial resolution as Landsat, and finally we have global scale. We use coarse spatial resolution data as MODIS. For the process, in general, we have to know that we have 7 steps to do the process. We will speak later about them in the part of methodology, and we will take this study as an example. Arrived to the transition zone, the last term, which represents the study area, is located between the Atlas-Tilian and Saharan. It constitutes the most extent part which narrows on the southern slope of the chain of Tilian Atlas, covers particularly the steppe zone, forming part of the high Algerian or Iranian plains. Administratively includes 13 townships and cover more than 60% of the total area of Tiert region. The climate is located in the semi-arid bioclimatic stage with cool winters, and for the vegetation is characterized by the vegetal formation of alpha and sparta. The major problem of the transition zone is the degradation of the steppe environment, and we can see this in the reduction of the pasture land in favor of agricultural land because of the pressure, anthropic pressures. So this caused cause the transformation of environment into heterogeneous environment, which cause difficulty in separating spectral response because of the overlap between the spectral properties, and that often leads to misclassification between several classes. So achieving precise and accurate classification depends on the characteristic of the transition zone, the availability and type of data, also the method of classification. So in the last decade, many algorithms have been developed to classify land cover, but the uprise of machine learning techniques has contributed to increase the precision. At the same time, the selection of an adequate spectral data to be integrated is important as the algorithm itself. In the case of multispectral sensors, it's usually preferable to synthesize the original spectral bands into indices related to the land cover trait as albedo or index water or vegetation index. So finally, the main goals of this study is to reconstruct the annual spatial distribution of land cover from 1984 to 2017. Also, develop a method for land cover mapping using only medium spatial resolution, capturing the particular spatial features in this region. The methodology regroups three parts: data acquisition and process, classification of land cover, and accuracy assessment. So first we have data acquisition. We choose ground truth data. We sampled 241 plots from the field and 83 points from Google Earth Probe. So the total 324 observation, a subset of 30% was set apart for validation purpose and the rest for training. As we can see here in this map, we found the 6 classes in the field: forest vegetation FV, steppe vegetation SV, agriculture and fallow land AFL, stable sand SS, unstable sand US, and bare soil BS. For the spectral data, we retrieved the 6 spectral reflectance band of Landsat 5 and 7 and 8 from spanning from June to September each year from Google Earth Engine. For the pre-processing, we use land render for the synthesize the spectral information into a single value per band and year, and after this we apply harmonization. Also, we calculated the NDVI, index water, color index, and Tesla capital transformation, and we select the T-mean as a texture indicator because the proposed methodology is to explore multiple combination of spectral data. After this, we extracted the complete spectral data of spectral information, and we apply supervector machine with 3 parameters. We choose the radial basis function, and we select cost and gamma. The model was trained and optimized using 70% of original data and tested with the remaining 30%. So the point was to combine spectral predictor from particular type and compare them with the counterpart that include textural indicator. And as we can see here in this table, we have the group 1, present 1 to the Landsat band, 3 for the Tesla capital transformation, and 5, 6 the index calculator. So to select the suitable model, we pass it to the third part, accuracy and assessment, where we calculate the percent accuracy, kappa coherence index, and the confusion matrix to summarize the classification accuracy between all the combination of observed and predicted. After that, we pass to the spatialization of 34 land cover maps. For the group of the Landsat band, we can see that the inclusion of texture parameters improve the classification. Also, the percent accuracy and kappa index for model 2 are much better than the results for the model 1. Also, we have no sandy soil classification errors, and the pixel confusion between steppe vegetation and agricultural and fallow land was reduced. For the group of tested cap, the results showed an increase in errors for classes of steppe vegetation, agriculture, and also stable sandy soil. And inclusion of texture parameters in the model 4 decreased the percent accuracy and added kappa index. Also, we have an increase in the confusion between several classes, except for the stable sandy soil classes. For the last group, the set of vegetation indices and the color indexes increase the classification errors mainly for the agriculture and fallow class with steppe vegetation and unstable sandy soil with stable sandy soil. In the model 6, the inclusion of the texture parameters with the set of vegetation index and the color index has increased the precision value and the kappa index. The model 2 was selected as the best model to classify the transition zone. So here we can say that the spectral bands in the Landsat time series gives a good classification result. And this is due to the different characteristic of the spectral bands, which increase the separability between different type of categories by reducing the classification error due to their sensitivity to change in the moisture content. This confirms their high potential in distinguishing different soil types. Or in the other side, the tested cap transformation showed a little sensitivity in the improving accuracy. Also, we found that the NDVI and index water are not suitable to use it in dry region due to the vegetation characteristic with the low moisture. Finally, we were able to produce 34 land cover maps with great precision corresponding to the annals of the transition zone between the Telian and the Saharan Atlas of the Cartesian. Also, this study allowed us to create an annual database and showed the importance of the supervector machine method in the classification of soils in the semi-arid regions. Thank you.
Thank you very much for your presentation. Clear and very much of results. So about the audience, if they have any question about the work, now listen. Any question, please? No question? So, okay, if there is no question, so we can start with the next presentation under the ID number 124. The number ID 124, I don't know if he or she is here. ID for that. Chair Boazas, if you have any question or intervention? No? So start quickly with the next presentation.
Good morning, everyone. In order to participate in the first international conference of the environmental biotechnology and biodiversity, we have the honor to present to you our work entitled Factors Affecting Soil Fauna Feeding Activity in the Two Sectors of Natal Head Park, done and presented by Nariman Khlayfia. The plan of our presentation is the following. First, an introduction. Second, materials and methods. Next, we have the results and discussion. And finally, we will see a conclusion. Now, global studies of soil biota have become the cutting edge of research in soil biology contribution to the diversity of abundance of nematodes, bacteria, fungi, insects, and earthworms. In order to understand the principle for sustainable agriculture, one must appreciate the significance of soil, more specifically, its biological diversity. The soil fauna and their feeding activity are considered indications of environmental change and soil quality, and therefore ecosystem functioning and sustainability. Given the complexity of the ecology of soil fauna, where the same group can function at different trophic levels, soil microfauna influence decomposition and biodegradation of plant and animal residues, as well as breaking down and redistribution of organic matter in the soil profile. That's why our work was aimed to study the effect of biotech and